Now, the generic transcription factors are the ones that are found in every close promoter region of every gene, because you need these generic transcription factors in order to flat out recruit RNA polymerase to the site. Most of our genes will have either Tata box or another type of promoter element right around the transcription start, which is, in this case, is the LNR sequence. Where the little pink arrow is, this is the point 0 of a particular gene, aka the transcription is initiated right there. And so the +10 and +30 is within within the mRNA, the transcribed part of the gene, and the -10, -30, -50, so forth, are things that are outside of the DNA template that will become mRNA, but they can contain a bunch of regulatory elements. For example, a Tata box will be within -30 of a particular types of promoters. And again, here's a Tata box binding protein here. And the words you can see on the slide outline the basic principle of how transcription factors, generic transcription factors, are able to recruit the RNA polymerase to a particular site. Now, before Tata binding protein and so forth can be recruited there first, it's the specific transcription factors that come in rearranged histones and so forth, something we don't have time to discuss in this class, but should be discussed in much greater detail if you take, say, MolBio 3000. So the basic idea here is that, say, if this is a Tata box containing promoter sequence in front of some sort of gene, and the first thing that happens is the Tata binding protein recognizes the Tata box and binds to the area, first recruiting TF transcription factor D, then A, and then the whole alphabet of transcription factors, resulting finally in recruitment of transcription factor H, which is the blue guy right here, which is an ATP-dependent helicase and actually separates the two DNA strands in that location, allowing for RNA polymerase to access the template that it will then transcribe into RNA. And once the H has bound and separated the two strands of DNA, allowing for transcription to occur, transcription begins, transcription proceeds from RNA polymerase 2. And this interesting thing right here is the tail that our RNA polymerase 2 has, and it's phosphorylated before RNA polymerase 2 can go. Again, extra details that might not be necessary for you guys to appreciate, but just to explain what the heck that part of the diagram was. And here we have a picture of what a basic bacterial promoter looks like and what a basic eukaryotic promoter looks like. Bacterial promoters are pretty simple. They will have the generic promoter site, which will contain a repressor binding site or an activator binding site. Repressors and activated are transcription factors which either prevent transcription or activate, facilitate transcription. Repressors, activators, easy. And then it's going to go through the coding region. In eukaryotes, you have in eukaryotes, you have a much bigger promoter complex, and usually the various activators that regulate gene expression in eukaryotes are very far away. They can be either upstream or before the gene begins, or downstream inside the introns of a gene here. And this is the first exon, and I'm assuming this is the first intron of the molecule. And I think the reason we have blue guys here for bacteria is not because bacteria contain introns, they don't, but because they're trying to depict here a polycistronic message, aka this particular gene cluster is actually coding for multiple genes, and this is just the termination sequences for one gene, then for another gene, then for a third gene, which is a very common approach in lots of bacteria. They have operons assembled into several genes assembled into operons. Eukaryotes don't do that. It's pretty much the rule, with probably very few, very rare and really strange exceptions when, as the rule, there's one promoter, one gene, one protein situation in eukaryotes. So let's talk about the generic promoter elements inside eukaryotes. Sorry about this confusing title. For eukaryotes, for RNA polymerase II, we have the Tata box we already discussed, and then sometimes we have the downstream core promoter element that can also affect recruitment of generic transcription factors that need to be there before RNA polymerase can go to the initiation element, the INR. And then enhancers or activator elements or specific transcription factor binding sites. These blue guys have lots of names depending on which year the book was written, literally, because somehow our terminology keeps on changing. These enhancer elements can be very far removed and are specific for that particular gene or that particular situation, and they don't necessarily they cannot themselves recruit RNA polymerase, but they affect the occupation, the availability of the DPE or the Tata box sequence. They make it visible to the generic Tata binding protein and the rest of the transcription factor assembly that needs to be there before RNA polymerase can sit down and go. So now let's look at how transcription factors actually bind DNA. Let's say this activated dude here is your generic transcription factor. And zoomed in, you would see something like this. You can see that transcription factors don't pry the DNA open. Only TF2H is able to pry the DNA open during transcription initiation. Instead, they interact the hydrogen bond. Oh, here we go. The hydrogen bond to the side chain, OH, hydroxyl, and hydrogen groups of the bases, because not every hydroxyl group of a nucleotide, of a nitrogenous base, is actually occupied when the DNA base pairs to self. There's still some nitrogens and hydrogens and hydroxyl groups present that can still interact with the outside environment. And you can see here's the same story as we saw in the past. If there will be some sort of charged amino acid that is able to form a hydrogen bond to the side hydroxyl groups protruding from the nitrogenous bases, depending on whether it's a guanine or an adenine or whatever, and that allows the transcription factor to stay in that particular location and then provide a binding surface for other proteins. And this helix-turn helix motif is a very common motif found in many transcription factors. That's just an aside. Don't need to remember that. And this, I just wanted to show you how this actually goes. So you can see here that we have, say, adenine and thymine together. And they have formed two hydrogen bonds to hold each other together. They base paired. But in the major groove, you remember how DNA has a major and a narrow minor groove? In the major groove and in the minor groove, there's still things protruding out that are charged and can act as hydrogen bond donor or acceptor, to which transcription factors can interact to, very specifically. And this combination of particular nucleotides in a row with their hydrogen bonds protruding on the outside allows transcription factors, specific transcription factors, but also the Tata box binding protein that specifically recognizes the TA sequence of the Tata box. It allows these proteins to interact with DNA in a much more specific fashion than, say, histones that we mentioned yesterday can, which just bind charged phosphodiester outsides of the DNA to spool the DNA onto these nucleosome spools.